For most of human history, a sudden fever or ruined crop felt like a curse. People watched their neighbors fall ill to sudden plagues, and their winter food supplies mysteriously rot away, with absolutely no idea what was attacking them. To make sense of the chaos, they relied on two main concepts. The first was miasma, the theory that diseases were caused by foul-smelling, poisonous air blowing in from swamps or decaying matter. The second was spontaneous generation, the belief that living creatures like maggots or molds simply materialized out of dead material. Because the human eye can only see objects down to about the width of a hair, we were blind to the microscopic forces actually driving these events. We were fighting a biological war with zero intelligence on the enemy. Without the physical tools to see smaller scales of reality, humanity was stuck treating superstition as science. In the 1600s, the first breakthrough came from an unlikely source. Antony van Leeuwenhoek was a Dutch cloth merchant who simply wanted a better way to inspect the weave and quality of his threads. He started grinding his own glass, creating tiny, highly precise lenses that could magnify objects up to 270 times their original size. Eventually, he pointed his lenses at drops of pond water and even the plaque scraped from his own teeth. Inside those seemingly empty drops, he found a bustling ecosystem. Thousands of wriggling, erratic shapes darted across his vision, tiny entities he called animalcules. This single observation destroyed the illusion of an empty environment. It proved an entirely hidden world existed right under our noses, raising a daunting question: what exactly are these invisible things doing? For the next century, scientists argued over the nature of these microbes. Skeptics like Francesco Redi and Lazzaro Spallanzani ran early experiments to argue that these tiny creatures had to reproduce like any other animal, rather than spawning out of thin air. In the 1860s, a French chemist named Louis Pasteur set out to settle the debate through rigorous experimental design. He boiled nutrient broth inside a custom piece of glassware featuring a long, curved, swan-like neck. The design allowed oxygen to flow freely into the flask, but heavy dust particles carrying airborne microbes got physically trapped in the curve. The broth remained sterile. Pasteur also noticed that when microbes were introduced to an environment, they performed active chemical labor. Specific types of bacteria were responsible for turning milk into cheese or fermenting grape juice into wine. By proving that microbes require parent organisms to reproduce, Pasteur stripped the magic away from microscopic life. He effectively exposed it as a mechanical, predictable biological system. If microbes were predictable biological machines capable of chemical work, researchers realized they might also be the engineers behind human disease. In the 1870s, a German doctor named Robert Koch took on the role of microscopic detective to catch these pathogens in the act. He started by examining anthrax, a lethal bacterial infection that was decimating European livestock. Under his microscope lens, he found a specific rod-shaped bacterium present in the blood of every sick animal. This flowchart visualizes the strict set of rules he developed, now known as Koch's postulates. To prove a microbe causes a disease, you must isolate it from a sick subject, grow it in a pure culture, and introduce it to a healthy subject to see if it causes the exact same illness. By mapping specific pathogens to specific illnesses, Koch birthed the germ theory of disease. This theory established that specific microscopic organisms are the direct cause of infections, turning a mysterious threat into a legible, conquerable battlefield. Armed with the germ theory, hospitals and doctors finally knew what they were fighting. They began implementing strict sanitation protocols, scrubbing away the invisible pathogens before they could infect vulnerable patients. Pasteur took this knowledge a step further. He learned how to weaken microbes in a lab, creating the first artificial biological shields, vaccines that protected humans against rabies and smallpox. In 1928, Alexander Fleming discovered that a specific type of mold produced a substance capable of destroying rogue bacteria. Soon, factories were mass-producing penicillin, the world's first antibiotic. With the invention of vaccines and antibiotics, humanity transitioned from helpless victims of sudden plagues to active combatants capable of clearing infections from our own bodies. Today, our relationship with the microscopic world has shifted. We have moved from simply identifying and killing pathogens to treating them as tools for intricate biological engineering. The Human Microbiome Project mapped the trillions of microbes living inside us. This community of bacteria, fungi, and viruses acts as a hidden parliament, governing everything from our digestion to our mood. Scientists like Jennifer Doudna and Emmanuelle Charpentier adapted a natural bacterial defense system into CRISPR, a genetic engineering tool that allows researchers to cut and edit DNA sequences with extreme precision. But the microbial kingdom is still adapting. The rise of antibiotic-resistant superbugs proves that these organisms are intelligent and constantly evolving to bypass our strongest medicines. The double-digit increase in global life expectancy since the 1800s began with a handful of people who stopped looking for cursed mists and started looking for biology. Their insistence on seeing the unseen provided the blueprint for every vaccine, antibiotic, and gene editing tool we use today.